Welcome back to my channel Knowledge Chronicle.
Today, we're diving into one of the most essential and intriguing chapters of Class 12 Biology: Genetic Disorders. This topic isn't just academic. It helps us understand how certain diseases run in families, how genetics plays a role in our health, and how scientists trace disorders across generations. In this video, we'll break everything down from pedigree analysis to Mendelian and chromosomal disorders. We'll use real-life examples and easy analogies so by the end, you'll not only understand every concept, but you'll remember it too. Let's begin. What are genetic disorders? Genetic disorders are diseases or conditions that are caused due to abnormalities in an individual's DNA. These abnormalities could be a mutation in a single gene, changes in multiple genes, or even abnormalities in the structure or number of chromosomes. Now, to study how these traits or disorders are passed down from generation to generation, scientists use something called pedigree analysis. Pedigree analysis, tracing traits through generations. So what is pedigree analysis? Since we can't control mating or do experimental crosses in humans the way Mendel did with pea plants, scientists rely on family history. Pedigree analysis is a tool where inheritance of a specific trait, abnormality, or disease is studied through a family tree across generations. Each individual in the pedigree is represented by symbols. A square represents a male, a circle represents a female. A shaded symbol shows the individual is affected. A horizontal line between male and female shows marriage. A vertical line leads to their offspring. Pedigree analysis helps determine whether a trait is dominant or recessive, whether it is autosomal or sex-linked, and who in the family might be carriers or at risk. Mendelian disorders are caused by mutations in a single gene. They follow the inheritance rules laid out by Gregor Mendel. Remember the laws of dominance, segregation, and independent assortment? These disorders can be autosomal dominant, where the trait appears in every generation. Autosomal recessive, where the trait appears when both copies of the gene are mutated, or sex-linked, especially X-linked recessive. Let's explore some key Mendelian disorders. Color blindness, when red is green. Suppose a man can't distinguish between red and green. That's color blindness, a sex-linked recessive disorder. It's caused by a mutation in the gene on the X chromosome, responsible for red or green cone cells in the eye. Since men have only one X chromosome, if it's defective, they're colorblind. Women, on the other hand, have two X chromosomes. So if one is defective and the other is normal, they're just carriers. About 8% of males and 0.4% of females are affected. If a carrier mother has a son, there's a 50% chance he'll be colorblind. Hemophilia, the royal disease. This disorder became famous because it affected Queen Victoria's royal family. Hemophilia is also X-linked recessive. It affects the body's ability to form blood clots. A small cut can lead to excessive, even life-threatening bleeding. A carrier female can pass this to her sons. Daughters are usually just carriers unless the father is also hemophilic, which is very rare. Sickle cell anemia, a curved twist in red blood cells. This one's an autosomal recessive disorder, caused by a mutation in the gene that produces beta-globin, part of hemoglobin in red blood cells. In this disorder, the amino acid glutamic acid is replaced by valine at the 6th position in the beta-globin chain. Due to a single base substitution, GAG becomes GUG. The result? The red blood cells become sickle-shaped, especially under low oxygen conditions. This leads to blockages in blood vessels, pain, and organ damage. Only individuals with two mutated copies, capital HB small hb plus capital HB small hb, show the disease. Carriers have one normal and one mutated gene and are symptom-free, but can pass the gene to offspring. Phenylketonuria, often abbreviated as PKU, is an inborn error of metabolism. It is also autosomal recessive. The problem? The person lacks an enzyme called phenylalanine hydroxylase. This enzyme normally converts phenylalanine into tyrosine. Without it, phenylalanine accumulates in the brain, leading to mental retardation. This excess also spills into the urine, which is why it's often diagnosed by a urine test in newborns. Thalassemia is another autosomal recessive blood disorder. It's different from sickle cell anemia. While sickle cell is about wrongly shaped hemoglobin, thalassemia is about not making enough of it. There are two types: alpha thalassemia, which affects alpha globin chains controlled by 4 genes on chromosome 16, and beta thalassemia, which affects beta globin chains controlled by 2 genes on chromosome 11. The more genes affected, the more severe the disease. Patients suffer from anemia, fatigue, and require regular blood transfusions. Chromosomal disorders. When the blueprint goes wrong. Unlike Mendelian disorders, chromosomal disorders are caused by abnormalities in chromosome number or structure. Let's understand the basic human karyotype. Humans have 46 chromosomes, 23 pairs. 22 pairs are autosomes. 1 pair is sex chromosomes, XX in females, XY in males. Chromosomal disorders arise from aneuploidy loss or gain of a chromosome. Polyploidy gain of a whole set, common in plants, rare in humans. Here are the most common examples. First, Down syndrome, the extra 21st. This is a classic example of trisomy, where there's an extra copy of chromosome 21. Discovered by Langdon Down in 1866. Features include short stature, a flat facial profile, a broad palm with a single crease, mental retardation, and delayed development. It happens due to non-disjunction during meiosis. Chromosomes fail to separate properly. Klinefelter's syndrome, a masculine-feminine mix. This condition affects males where instead of having the usual XY chromosomes, they have an extra X, making it XXY with 47 chromosomes. The features include a male appearance, underdeveloped testes, gynecomastia or breast development, and sterility. This condition is due to non-disjunction, where the sex chromosomes fail to separate properly. Turner's syndrome, the missing X. This one affects females who have only one X chromosome, XO 45 chromosomes. Features include sterility due to rudimentary ovaries, short stature, and lack of secondary sexual characters. Unlike Klinefelter's, Turner's patients have no Y chromosome. Let's quickly revise what we've learned. Pedigree analysis helps trace inheritance patterns. Mendelian disorders are caused by single gene mutations. They can be dominant, recessive, autosomal, or sex-linked. Chromosomal disorders involve gain or loss of chromosomes and lead to syndromes like Down's, Turner's, and Klinefelter's. Understanding these disorders not only deepens our knowledge of biology, but also increases awareness about genetic counseling, early diagnosis, and potential treatments. If you found this video helpful, imagine how many others would benefit from it too. Your one like, share, and subscribe will help us create more valuable content. So don't forget to click that button and support Knowledge Chronicle. See you in the next episode, where science comes alive.